you so much. Thanks for the invitation. A little bit of a switch of gear now to something about MRI techniques, some of the techniques that we've heard mentioned before for probing brain iron and neurodegeneration. Just a little overview of what I'm going to talk about today. So we'll talk about this technique called quantitative susceptibility mapping, which is quite a mouthful, so I'll call it QSM for short. I'll talk a little bit about its reproducibility and robustness, some work we've done, and also, of course, some clinical applications, but coming from a more physics-based point of view with our clinical collaborators. And then finally, I'll mention a newer technique called susceptibility source separation. So what is QSM and how do we do it? Yeah, there we go. It's just a bit of a delay. Right, so magnetic susceptibility is an intrinsic property of materials, and it determines how that material is going to behave when you put it inside the big strong magnetic field inside the MRI scanner. So we have this spectrum of susceptibilities across the, I don't know if we have a mouse here, but yeah, there it is. I saw something. Yeah. Across the different materials in the body, and you can see that there's calcifications and other materials that are diamagnetic. That means they have a negative susceptibility relative to soft tissue. And there's also materials like ferritin and deoxygenated blood that are paramagnetic, and they have a strong positive susceptibility, so they concentrate the magnetic field inside them. And some of the physical origins behind that, so the diamagnetism is much weaker, and that arises from the sort of field-induced alteration of the electron orbits around the nuclei, and that's independent of temperature. And then the more dominant effect, and the effect that we're talking about with respect to iron, is paramagnetism. That's the positive susceptibility, and that arises from these unpaired electrons that align, their electron spins then align with the main magnetic field inside the scanner, and that's much stronger, as I said, and that increases with temperature. Increases as temperature decreases. So that's a little bit about the physical origins behind the effects that we're seeing. And you can see that in the human brain, there are several different sources of magnetic susceptibility. So it's easy to say that with susceptibility, we're quantifying brain iron, but actually that's not the case. There are many different contributions to susceptibility, and we'll see that. But some of them include, obviously, ferritin, which is strongly paramagnetic, and also myelin, which we believe to be weakly diamagnetic as well. And as I mentioned, calcifications and other things. So these are just some of the contributions that we see in the human brain. So how can we probe this magnetic susceptibility in MRI? So if we knew the underlying susceptibility distribution in the brain, then we could do this calculation where we convolve it with this dipole distribution, and we could calculate the field perturbations that we would observe. And that we could then measure using the phase of the MRI signal, which I'll explain in a moment. So that would be great if we knew this a priori, but in fact what we're trying to do is go the other way. So we're going to measure the MRI phase and try to calculate the underlying tissue magnetic susceptibility distribution. So what is the phase in MRI? Well, in standard MRI, we have this magnitude signal. So we measure the length of this magnetization vector in the transverse plane. And that gives us the images that you will all be very familiar with, these magnitude MRI images. But this magnetization vector also has an angle as it processes in the transverse plane. And we can measure that angle. That's the phase angle. And we can also create phase images. And you can see that those images look quite different. They have, for a start, they have these wraps, and that's because the phase is an angle that goes round and round, and every time it crosses this axis, then it jumps by from 0 to 360 degrees or 0 to 2 pi, and so you get these wraps that we have to remove as one of our processing steps. But the good thing is that the phase is directly proportional to these field perturbations that are caused by changes in the magnetic susceptibility. So that's what we're really, really interested in here. So we might say, okay, great, we can measure phase images, we can just stop there, that's fantastic, that's all we have to do. But actually, if you look at the phase, it's affected by both the geometry and orientation of the tissue with respect to the main magnetic field inside the scanner. So if you look, for example, at this piece of fixed human brain tissue, you can see that when it's roughly parallel to the main field, it looks bright. and when it's roughly perpendicular to the main field, it looks dark. So this isn't very useful if we're looking for something that's consistent, that has a consistent contrast in this layer in the brain. You can see the same thing here in the fixed rat brain, where depending on its orientation, then you get very different contrast in the white matter and the corpus callosum here. So, the phase images they are sensitive to changes in the magnetic susceptibility, but they have this orientation dependence, so much less useful. Also, if we look at the phase differences outside, for example, the red nucleus and substantia nigra in the human brain here, then you can see that the phase differences actually take place outside those structures. So the phase changes are non-local. They have this kind of dipolar effect that extend beyond the structures. And so that's not very useful if, for example, if we want to target a structure for deep brain stimulation or something like that. So we really need something that has local contrast and that doesn't depend on the orientation. And so that's why we want to calculate the underlying tissue magnetic susceptibility distribution from the phase. So how do we do it? First thing we need to do is we need to acquire the right kind of MR image, and that's something called a T2 star weighted gradient echo image. And that gives us, we then need to make sure that we save the phase, right? So often, because MRI scanners are set up to output the magnitude signal, people often throw away the phase, and then we can't do anything about it. So if you're interested in calculating QSM to look at brain iron and other compounds, then the first thing I would say is make sure that you're not throwing away the phase information. The second thing we need to do is remove these nasty wraps that we saw before, and there's plenty of algorithms to do that, and that gives us a much nicer, smoother image. We'll get rid of these 2 pi phase jumps. You can see that this image is dominated by the difference in susceptibility between tissue and air. So you can see, for example, these very large field perturbations or phase differences around the auditory air spaces. And so we want to remove those large scale, what we call background fields, because those aren't the small field perturbations that are caused by the susceptibility differences in the tissue. And so the next thing we need to do is remove the background fields. And again, there's a whole host of algorithms for doing this, and I'll tell you a little bit about some of the consensus that we've reached about how to do this. So then we've got our local field perturbations, and the next step in this QSM pipeline, if you like, is to take those and solve an inverse problem. It's an inverse problem because we're going from something that we've measured to the underlying signal source, which is susceptibility. And again, there are lots of different ways of doing this now. But then you can start to see that we can, again, see the positive paramagnetic susceptibility in the deep brain grey matter, which is very iron-rich. So you can start to see the kind of typical QSM map or susceptibility map. So that gives us our output susceptibility map. And the good thing about this is it does indeed overcome the non-local phase contrast. So if you look now, this is one of the first images from the very first QSM paper in 2009, so it looks a bit noisy. We've got cleaner images now, but the point is the same, is that around these structures, you no longer see the changes happening outside those structures, but the susceptibility changes inside those structures. They're local. So we've got now a very nice local contrast. The other thing, the other advantage of susceptibility maps is they overcome that orientation dependence that we saw. So here in this piece of fixed brain tissue, if you look at the phase images, you can see that the contrasts in these layers are almost opposite when you rotate that piece of tissue with respect to the main field. But if you look at susceptibility maps, they look much, much more similar. And we can quantify this, and you can look more closely. But also, if we draw profiles across some of these structures, you can see that the contrast in the phase images is almost opposite, but the contrast in the susceptibility maps is almost 100% similar. So that's resolved that non-local orientation-dependent contrast now, and we have a contrast that is not dependent on the orientation of the tissue with respect to the main magnetic field. So as I mentioned, we've got lots of different sources that contribute to the magnetic susceptibility, from ferritin, which is the one that we're probably most interested in, to deoxyhemoglobin, which is very strongly paramagnetic, myelin, calcifications, inflammation. And this leads QSM to having many different clinical applications, from targeting structures for DBS, looking at MS, Parkinson's, Alzheimer's. I'll show some work we've done in sickle cell anemia. We can also look at microbleeds in Alzheimer's and other diseases. We can look at hemorrhages, demyelination, epilepsy, and also inflammation in active rim lesions in MS. So there's a whole host of applications. And the other great thing about doing QSM is that when we acquire this kind of data, and we save the magnitude in the phase, it's very efficient. We can squeeze a lot of information out of it. So we've got our standard magnitude image that is very useful, and we've also got our phase image, and we can calculate the decay time, the R2 star, which tells us how quickly our signal decays, and I'll show you another use for that later on. We can also, this is very clinically widespread, we can do something called SWI, susceptibility weighted imaging, where we multiply the magnitude image by a processed version of the phase image, and that amplifies the phase changes from something like microbleeds. And so that's it's used a lot to diagnose traumatic brain injury and find microbleeds and hemorrhages in the brain. So it's very popular. But we can also, of course, do our QSM reconstruction and get our susceptibility map, which gives you these advantages of overcoming the non-local contrast and the orientation-dependent contrast. Okay, so how repeatable and robust is this technique? The good thing is, because I said there's now because this technique's been around for over 10 years now, almost 15 years, I guess, then there's a whole host of algorithms for every stage in that reconstruction pipeline. And so our community came together, the electromagnetic tissue properties mapping community of the MRI world, came together and decided that we needed some consensus to help people to use these techniques, to not have to navigate through the zoo of algorithms. And so we've recommended now techniques for use in the brain. And so you can go to this paper and kind of see from beginning to end how to how to acquire these images, how to process the images, and how to present them as well. So this is a really useful paper if you want to get into doing QSM. And one of the recommendations of this paper is to reference the results that you get in every voxel to a reference region, because the thinking behind that is that the overall susceptibility in the brain might change in disease or with age. As we saw, the iron levels increase with age. So there was thinking that we should reference our susceptibility. And we've done some work to figure out what the statistical impact of this referencing is on the QSM. And here's some of the reference regions that have been suggested as having very stable susceptibility. You can see here we've got the ventricles, so the cerebrospinal fluid. We've got some white matter regions like the corpus callosum and internal capsule, and also regions that have a very long R2 star, and also a very small variance in their signal. So there's different ways of defining those regions, those reference regions. And so again, the goal of this is to reduce inter-scan variability and inter-subject variability. And that's because we can't define an absolute susceptibility reference. All the values that we get from QSM are relative because of the way that the algorithm works. And we define referencing as subtracting the mean value from one of these regions from every voxel. That's how we define it. And this figure shows for all different kinds of QSM algorithms, this was taken from the QSM challenge where we tested lots of different algorithms against each other, and using different reference regions. And the squares that are filled in are those that are that don't have a susceptibility that's significantly different from the ground truth. This was done in a phantom where we knew the underlying susceptibility values, so we could test this. And what you can see is that if you use something like the corpus callosum, that actually gives you less accurate results for many of even the top scoring algorithms. You've got less squares that have accurate susceptibility values. And so actually, it may not be a good idea to use that as a reference region. And here again, you can see now the statistical impact of referencing in a study of epilepsy, which I'm going to show later on. And you can see that if you use, for example, the CSF as a reference region, then you get less significant differences than with no referencing in comparing the groups, for example, of left and right temporal lobe epilepsy groups. So actually, choosing CSF reduces your effect size, and you might get more false negatives or fewer true positives. But if you choose the corpus callosum as a reference, you're going to get more significant regions of interest. And that's because the corpus callosum actually already has between group differences between the different groups. So it's not a good reference region to choose. And this happens because if you do the referencing, it actually propagates the variance into your referenced ROI value. And we can see this happening here. What we're plotting here is the difference in the variance after referencing compared to without any referencing. And you can see that, for example, if you use the CSF as a reference, again, you get this decreased variance in your region of interest, and you're going to get an increased effect size when you're comparing different groups. So actually what we recommend, which is a bit against the consensus, but we think it's better not to use an explicit reference region because actually from the way that we do the calculation, the results are kind of implicitly referenced to the whole brain anyway. So we don't need to actually do another kind of explicit referencing which just adds variance to our study. And this is safe to use the whole brain kind of implicit referencing that's already built into the algorithm because it won't increase your number of false positives. It's not going to change your ROI variance. So that's just coming out in Magma. Okay, we wanted to know how repeatable doing all of this QSM is. And there have been some studies on QSM repeatability. And here what you can see is that we've compared several different state-of-the-art QSM algorithms in the whole head and neck, actually, because we're interested in not just the brain, but obviously this is a brain meeting, so I'll just talk about the brain here. But you can see that these look very, very different, the contrast of these maps in the brain. And these are the susceptibility values in the deep brain grey matter regions of interest from the different algorithms. And what you can see here are the significant differences. So these are ANOVA values. And the only values that were not significantly different when using two different methods were these orange ones. So actually, the methods that you use makes a huge difference. But, so that's the main message, is that the method makes a huge difference, which we've shown that before. But we still get repeatable results, but you need to just make sure that you choose the right method. And the question that we had was which repeatability metrics are best? And if you look at the across subject variations, so repeated session variations, and also the intra-class correlation coefficient, they show you that this method called auto-NDI, which is a method that we've developed for QSM, gives you the most repeatable susceptibility values. That's I think the purple one over here. And that these are the best metrics. We tried a whole bunch of repeatability metrics, but these are the ones that really illustrate the repeatability the best. So that's something, again, that's just coming out in a special issue of Magma. All right, so moving on to the clinical applications of QSM in neurodegeneration. I'm going to start off by talking a little bit about cerebral amyloid angiopathy. So one of the good things about QSM is that it can, unlike just phase images or SWIs, it can actually resolve or tell you the difference between positive susceptibilities and negative susceptibilities. So we can, for example, distinguish a calcification from a bleed on the basis of their susceptibility, their appearance in the susceptibility map. And we applied this in patients with cerebral amyloid angiopathy who have this whole host of hemorrhages and microbleeds. And one of the interesting things you can see here is that sometimes clinically, the clinicians like to count microbleeds to kind of quantify the severity of these conditions or look at aging or other kind of diseases, especially it is now becoming important in the treatment of Alzheimer's with the new drugs whose names are escaping me at the moment, and so we can start to monitor ARIA by counting microbleeds and things like that. And what you see in the susceptibility weighted image, which is based on the magnitude image and the phase image, is that you actually see some of the microbleeds seem to disappear in the quantitative susceptibility map. So if you counted it on this image, you might get a worse score. And this is because of this dipolar effect. So if if you're counting the phase effects, you can see that there are these kind of two blobs around susceptibility source. So you might be counting the microbleeds twice, whereas in the susceptibility, you've just got this single source that you're counting. We're trying to quantify this, but the good thing is that you can differentiate these things based on sign, and also you can when it's just single sources, you can do some quantification and try to determine the concentration of iron in that microbleed. We've also looked at Parkinson's disease, and some of the work that I've done with Professor Ramona Weil and Mina Wrighton and George Thomas, we're interested in why some brain regions are more affected in Parkinson's disease, and we've looked for relationships between previously observed changes in susceptibility in the brains of people with Parkinson's disease and gene expression in those same regions in the healthy brain. So this was correlating gene expression in the Allen Brain Atlas with susceptibility changes that we found in the brains of people with Parkinson's. So here are the kind of correlations between whether people had Parkinson's and QSM, the QSM changes in Parkinson's, and here are the sort of mRNA library at the Allen Brain Atlas. And when you correlate those things using a partial least squares regression, then you can look at which genes are expressed in those regions that are correlated, and also what kind of cell types are being expressed there. So what was found is that there were indeed these correlations, and that the genes that were implicated were genes indeed for heavy metal processing, including metal ions, presumably iron as well, and these relate to the regional susceptibility increases, and also synaptic signaling genes as well. So we get this association with a distinct gene expression profile, and this might give us some information about why these brain regions are the most affected, and perhaps suggest also future targets for slowing the progression of neurodegeneration in Parkinson's. Looking briefly at multiple sclerosis, there's been a huge body of work on QSM in MS, so I'm not going to review it all. There's a very nice review that came out in 24, and they've got some main findings, which is that compared to controls, QSM values are increased in the basal ganglia, so deep brain grey matter, but reduced in the thalamus. The susceptibility values in white matter lesions change over time. They increase first, and they plateau, and then they decline. And there's a whole study now of these paramagnetic rims of these lesions as well. So that's a whole other area. And the other thing to observe is that there's a lot of difference in the methods that were used to acquire the data. So there's a lot of harmonization work that needs to go on to try and kind of synthesize these results together. A study that I was involved with at UCL was looking at lower thalamic susceptibility and disease severity in secondary progressive MS. So the questions that they were trying to answer was, are there abnormalities in susceptibility in an older progressive MS cohort? Does it correlate with disability? And this was baseline data from a trial. So you've got the numbers here. And what we found was that this reduced susceptibility in the thalamus in these people with SPMS suggested a lower iron content, and that was associated with more severe physical, cognitive, and visual disability. So again, suggestions that for the thalamic iron might correlate with important mechanisms in the clinical disease progression. Now moving on to temporal lobe epilepsy, the study that I mentioned before. And just to show you that often, so we now have these consensus methods, but most of these studies were done before we kind of converged on the consensus. And sometimes you still have to optimize your QSM pipeline for the particular data and study that you have to get the best out of your results. And here's an example of that. So using our kind of standard method, you can see that there are these dark regions that propagate through your susceptibility map. And so we developed, used a method that had been developed by someone in our group called weak harmonic QSM to remove those kind of residual background fields and give us a more homogeneous contrast throughout the brain. Here you can see that we got significant susceptibility differences in several regions. You might expect, because these people had hippocampal sclerosis, many of them, so we did find a significant change in susceptibility in the hippocampus in the left temporal epilepsy group compared to healthy controls. There were also changes in the ipsilateral thalamus, and also bilaterally in the putamen, so increases in susceptibility in the putamen relative to healthy controls. And this suggests, again, increased iron content, and it does reflect disease severity again. So that's in temporal lobe epilepsy. We also looked at focal cortical dysplasia lesions, and it's a different group. These are children with FCD, and we looked now at the inside the cortical layers in the brain, and found actually that the sort of susceptibility distribution that you get in the healthy homotopic regions on the other side of the brain was kind of disrupted in the lesions. You can see the significant difference between the susceptibility at different cortical depths in the lesions compared to the homotopic regions on the other side of the brain. So there seems to be a breakdown of the normal cortical structure that's reflected in the susceptibility maps as well. And comparing susceptibility maps now with histology, with staining, you can see there were changes in calcium and zinc, so increases in calcium and zinc in these lesions, and actually decreases in the iron content in these lesions. So we can see that, again, that susceptibility is sensitive to all these different changes in all these different elements or molecules, and is associated with these kind of histological changes. So getting the histology alongside the QSM gives us insights into what's causing the susceptibility changes. Okay, and finally, showing you some work we've done in people with sickle cell anemia. This is my collaborator, pediatric neurologist Fenella Kirkham in London, and also neurologist Mboka Jacob in Tanzania, in Dar es Salaam. And as many of you know better than I do, sickle cell anemia is a genetic disorder affecting hemoglobin, and the blood cells become sickled, and they have this abnormal shape, and they can't carry enough oxygen. And you get all sorts of horrible symptoms, including vasculopathies, strokes, silent cerebral infarcts, cognitive impairment, all kinds of very bad pain crises. And we also know that brain iron is very important in cognition, as we've seen, both at the beginning of life and at the end of life in dementias. And we know that iron overload may also accelerate neurodegeneration. So, and because we're using, QSM often as a proxy for iron as well as other things, then we thought we wanted to use QSM to see what might be going on in the brains of these people with sickle cell anemia. So here's a whole brain analysis, this time not restricted to specific regions, but a voxel-wise analysis. And we can see increases in susceptibility in many deep brain gray regions, and also some decreases in susceptibility in white matter regions. But we also see, what's quite interesting is we see a faster increase in susceptibility with age in these children and young adults in people with sickle cell anemia than in healthy controls. And that is looking at some of the specific gray matter, deep brain gray matter regions. And these are data from the UK, but we also have analogous data from Dar es Salaam, and it'll be interesting to see what happens because that's in the context of iron deficiency. So it'll be very interesting to kind of compare those data when they're through. We also looked at the susceptibility in the superior sagittal sinus to see if there was an overall change in brain oxygenation. Remember, I told you that deoxyhemoglobin is strongly paramagnetic. So in fact, we do see that. We see this we see a significant decrease in oxygenation in venous oxygenation in people with sickle cell anemia in the superior sagittal sinus. So there's this major change in their overall blood oxygenation. And this is completely non-invasive, so it's, it's really useful, hopefully. And also, if we can use a model called QQ, which is combining quantitative BOLD and QSM. It's all the same data that we've acquired, but we're now putting it into a model of oxygenation in tissue, not just in vessels. And we can use this model to see a significant difference, a significant increase in the oxygen extraction fraction in sickle cell anemia. And that's obviously because they're trying to kind of pull as much oxygen as they can out of the blood because there's not that much there. So this is really interesting, and we're looking into investigating this further. And finally, as Ashley mentioned, there's increases in iron in lots of different, in aging, but there's also, and in lots of different diseases, there's changes in magnetic susceptibility in a whole host of neurodegenerative diseases. And so here's some of them, a review of some of them, and just giving you a flavor of some of the ones that we've worked on. But so this is a tool that is being applied in many, many different neurodegenerative diseases. But the thing is, there are lots of different processes going on in neurodegeneration. For example, demyelination and ferritin increases. And so it would be really useful if we could disentangle those sources of susceptibility, because sometimes those changes are happening in the same voxel. So we might have increases in ferritin, which is a paramagnetic source. We might then have demyelination in the same voxel. And the question is, can we disentangle those effects? If we just use susceptibility, then we might see things cancelling out, or things going in the same direction, but caused by different sources. So if we look at the R2 prime, that will give us a kind of, is proportional to the sum of all the susceptibility sources, irrespective of whether they're positive or negative in that voxel. And so recently there have been methods developed called susceptibility source separation, which allow us to combine the R2 and R2 star information that we get from the magnitude signal decay with our susceptibility information to separate the paramagnetic and diamagnetic sources. And so again, this is what I was saying, they can happen in the same voxel, and it would be really interesting to kind of separate those results. This is just showing you two of the models that are out there. There are now a few of them out there, so some of the maths behind you that I just showed in a very simple form. And these are the results that they got. So you can see that there's actually quite different maps. You can see it probably better here. The positive susceptibilities and the negative susceptibilities, but sometimes in the same voxels. Okay, so it's not just like that you can split the QSM map into positive and negative values, but those changes are happening in the same voxels. And those correspond quite well, especially in these deep brain grey matter regions, with the iron and myelin. So we actually looked at 7 available methods for source separation, and we compared them in a healthy human brain. There isn't a gold standard. Unfortunately, we don't have, this is how much positive susceptibility and this is how much negative susceptibility. But we had a look, and we tried to see how much source separation they gave us. So we acquired these, and we found that actually this one, the chi separation with MEDI and with R2 information included, gives us a really nice separation visually between the positive susceptibilities and the negative susceptibilities. So they're not close together, like in this case where they're very faint. And then in this case they're pretty much all positive, so something's gone wrong with this algorithm. And also this method seemed to have quite good repeatability. We did several repeated scans, had good repeatability compared to the other methods. And you can see that quantified in a few different regions of interest here. So here you can see that these methods had pretty small source separation. And these ones had high source separation. And again, these ones were kind of all positive. So obviously something's gone a bit wrong there. Finally, I'm just going to go through what I hope I've taught you in this short talk, that QSM is an efficient technique, and it overcomes the disadvantages of phase and SWI contrast. It's a highly repeatable method in the brain, but the results that you get are going to depend on the method that you use. But now there's recommendations, so you can see what to do. As I said, recommendations. So if you do reference, in addition to kind of the implicit referencing that's there in most of the methods, then that is going to propagate uncertainty and kind of might increase the number of false positives that you get in your study. So we recommend actually not explicitly referencing. You can see that this is now being applied in several different diseases to detect changes in susceptibility, not just due to iron, but due to many other different contributions from myelin, calcifications, venous oxygenation, and now even OEF if you use the QQ model. And now we can add to QSM by doing source separation, which is helpful in providing maps of paramagnetic and diamagnetic sources. And we can improve those by including the R2 information as well. I'd really like to thank my group who worked really hard to provide all this data, my funders, and I'd also like to thank all the collaborators that we've worked with. We couldn't do all this without the clinical collaborators that we work with as well. So thanks very much for your attention.